With a solid conceptual understanding of what dimensionality reduction is, we're going to work through two conceptual examples here, moving from two variables that we talked about in the last video to working with more and more features. And we want a better understanding of what scree plots are, which I mentioned but didn't really give you a full understanding in the last video, and further understanding about loadings. So these data I started to introduce in the last video, but didn't go into tons of detail. So I showed this plot of two features talking about stock prices and showed that if the scatter plot between these two features, and you wanted to draw principal component 1, that would be the vector that explained the most variance in the data, and principal component 2 would be the vector that is orthogonal to PC1 that explains the second most variance in the data. And this was for two stocks, Chevron and ExxonMobil. And we talked about the process of calculating the linear combination across all features and doing this for this number of features you have in your data set, each of which calculates a principal component, and you retain as many components as needed. But I didn't talk about really how to do that and what it means to retain as many as are needed. So let's move beyond just 2 or 3 features to, say, a data set where you have information across time. So these are data from 1993 to 2015. I'm just showing a snapshot of this data set here. Our features are different stocks. So we have 517 different stocks in this data set. But let's just for now focus on the top 16 companies. So we're going to have 16 features and a whole bunch of observations across 5,000 plus days. If we were to generate PCs from these data, each component would have an associated eigenvalue, with the first principal component having the largest eigenvalue. And so what we're looking at here are eigenvalues on the y-axis and then the first 10 principal components on the x-axis. So this is a scree plot. So when we're talking about scree plots, we're talking about the eigenvalues and for each different component. The vernacular definition of scree is an accumulation of loose stones or rocky debris lying on a slope or at the base of a hill or a cliff. And the reason this is called a scree plot is because we're looking for the place where that rock would just kind of tumble down to a flat. So often we talk about looking for the elbow in this plot where most of the variance explained happens, and then it kind of just drops off to nothing. So this happens here around 4 or maybe around 7. And this is how visually when you're doing EDA, you know how many principal components to consider. When we get down to 7, 8, 9, and 10, these are explaining almost none of the variance. So we've taken our 16-dimensional data set, and we can really focus in just on these first 3 principal components as they explain most of the variance in your data set. So in a scree plot, it's desired to find a sharp reduction in the size of eigenvalues. So we're looking for the cliff. Now, maybe we just care about the first principal component, but there's a drop off here between 3 and 4. So when the eigenvalues drop dramatically in size, adding an additional principal component would add relatively little information to that we've already extracted. Remember, each principal component is a linear combination of all features. So we're considering all of the data in each of these principal components. So we want to take those principal components that explain the most variance in our data. When we look at what that means under the hood and we look at the component loadings, and that's what we have on the y-axis here, across our 16 top companies, we can start to see in each principal component what the loading is for each specific stock. So we can see that we have negative loadings across all of the stocks in the first principal component, but we start to see variability so that some stocks have positive loadings and some stocks have negative loadings, and these are determined when you calculate the principal components. And we, despite not telling PCA anything other than the stock prices, can start to see what each principal component represents. Remember, each component is comprised of information across all of your features. So we can see that PC1 is the overall stock market trend. PC2 is the price change of energy stocks. So if you start to look which stocks are positive here relative to the other ones, these are going to be our energy stocks like ExxonMobil. PC3 starts to capture the movements of Apple and Costco. PC4 is the movements of Schlumberger to the other stocks, right? There are a few that stick out in PC4 relative to all of the other stocks. So this Schlumberger is driving the variance we see in component 4. And PC5 is really driven by financial companies. So the differences in those stocks relative to all others are driving the loadings we see in PC5. So there was no label given to each of these principal components beforehand. The computer calculated the principal components based on the data that were there, but after the fact, we can see what information is being distilled at each PC. Now, you can't always do this in your data and pick out the trends, but often this is how you start to understand the relationships between the variables in your data. Now the question is how many PCs to select. So one is you visually look at the scree plot, look at the place where you see the cliff. So maybe you would consider the top 3 principal components here, maybe the first one. But you can also look at the percent variance explained. So maybe you want the principal components that explain 80% of the variance in your underlying data. Option 3, you can inspect the loadings for an intuitive interpretation like we did in the last slide. And option 3 is you can use cross-validation to see how many principal components to include. All right, so key ideas here. Don't forget PCs are linear combinations of the predictor variables and the input data has to be continuous. They're calculated to minimize correlation between components. This is going to reduce and minimize redundancy. Some limited number of components will typically explain most of the variance in the outcome variable, so you only need to use a few principal components, regardless of how many input variables you had. And this limited set of PCs can be used in place of original predictors. This is the point of dimensionality reduction. So for example, you might end up with something like this at the end of your data set. So these are biological data from a mouse experiment, where each point is a mouse and each position is its relationship between PC1 on the x-axis and PC2 on the y-axis. And the points are colored by their group, their experimental group, but those groupings had nothing to do with how the principal components were calculated. It's just after the fact. And we can see that groups 2 and 4, orange and green, are most similar to one another because they group together, where group 1 is somewhere between group 3 and our groupings over here, and that group 3 is the most distinct. So you can start to get the overall relationship across the underlying data. So plotting the top principal components can reveal groupings within your data. And a scree plot can help identify how many PCs to consider. So you're going to look for that elbow. And we can see PC1 and 2 in these data really explain the vast majority of the variance in the data, and then it kind of drops off starting with PC3. A loadings plot can be helpful as it projects the values of each PC onto how much weight they have on that principal component. So we can start to see that NPC2, and it doesn't matter what the underlying data are here too, and CHIT1 strongly influence PC1 as they're the furthest values there, while our y-axis here is principal component 2, and here that's where we're looking at GBA, and LCAT start to have more say in principal component 2. So these loading plots can help to see which features from your original data set are driving the differences you see in a particular component. All right, so with that said, let's talk about these examples. So our case study here is diet in the UK, and the underlying data are 17 foods across 4 countries. And you can envision this having way more foods and way more countries, but we're just going to focus on these 4 here. And you see information about their relative consumption in each location. You can calculate principal components from these data. And what you do when you look at PC1, you then see that Wales, England, and Scotland tend to cluster together. And you can already, in a single variable, using all these underlying food information as your inputs, see that Northern Ireland is distinct. If you take into account principal component 1 and principal component 2, you see clear separation between Wales, England, and Scotland and Northern Ireland. If we look back at the raw data, Northern Ireland eats way more fresh potatoes and way fewer fresh fruits, cheese, and fish. And this reflects real-world geography. So if I were to ask you, why do we see the separation? We can see England, Wales, and Scotland are connected and have shared borders, whereas Northern Ireland is separated by a body of water. Right, so this geographic information was not an input to the model. It was just the number of foods, but principal component analysis started to recapitulate this geographic distribution, where we see Scotland, England, and Wales being separated from Northern Ireland. We see this beyond food there's this very famous paper from many years ago now called Genes Mirror Geography within Europe. And what these geneticists did here was they took data from some 1,400 people and they looked at, say, 500,000 SNPs. It's not super important what a SNP is, but what you need to know here is your DNA in all your cells is made up of G's, C's, T's, and A's, these 4 bases. And a SNP is a position in one's DNA that varies between individuals. So for example, maybe this purple human has an A, this blue human has a G, and this orange person has a T. This would be a SNP. They're similar throughout much of their DNA, but there are going to be places where you and other humans differ. This is what results in normal human variation. And this is what things like 23andMe and Ancestry.com use to determine your genetic ancestry. So they used these data, they measured these genotypes, these SNPs, at 500,000 different places and a whole bunch of people. They calculated the principal components from these 500,000 features. And then they just looked at principal component 1 and principal component 2, where each point on this plot is an individual. And they compared these results to the map of Europe. And what they saw, where each person here is a different individual, the letter here has to do with their geographic ethnicity. So DE here would be Germany. And we can see the position on PC1 and PC2 recapitulates the map of Europe. So just by taking 2 features out from these 500,000 different features they started with, they can then generate the geographic relationships underlying the genetic ancestry in these individuals. This goes beyond to then start capturing information about linguistic differences and north-south differences in who speaks what language. So it can get very, very nuanced just by looking at a few principal components. All right. So with those 2 examples showing how you can use diet and how you can use genetic information to take a whole bunch of features down to a few principal components, these are places where if you need to compress data and reduce storage space, you need to reduce computation time, so you only have a few features rather than hundreds or thousands. You want to remove redundancy and identify outliers in your data. That's where principal component analysis can really shine. Now, it does lead to some data loss. When you go from 100,000 features down to 2, you're going to lose some information. It tends to find linear combinations between variables, and sometimes the information isn't a linear combination, so it might not be the right model. It's going to fail in cases where mean and covariance are not enough to define the data sets because that's what it's using under the hood. And you may not know how many principal components to keep, and it is highly affected by outliers in the data. So if you have a few outlier points, it's going to affect your principal components calculated. All right, that is the end of our conceptual understanding, and jump into a quick Python example next.